Welcome to the webinar, Bridging In Vitro Data to Clinical Outcomes with MetID. My name is Michael Milholland, and I am the Global Marketing Manager here at XenoTech. Today, I am joined by Heather Heading, Director of LCMS at Agilex Biolabs, and Dr. Andrew G. Taylor, Manager of Technical Support for Services at XenoTech. Heather obtained her Bachelor of Science with honors and a Bachelor of Technology from the world-renowned Flinders University in South Australia, where she then joined Agilex Biolabs in 2009. Now Heather is responsible for the management and oversight of Agilex Biolabs LCMS division and works closely across the various departments to ensure her clients are provided with an exceptional customer experience. She is passionate about bringing new medicine to market for a healthier world and sees the crucial role that her work with Agilex Biolabs does as a vital part of this process. Andrew received his Ph.D. from the University of California, San Diego. He joined XenoTech as a research scientist in 2007, serving as a study director in non-clinical drug interaction contract studies in compliance with good laboratory practices, OECD Principles of GLP, and Japan MOHW GLP Standards, and specializing in drug transport and drug metabolism studies. He became the Technical Support Manager for Services in 2020 and provides valuable guidance to ensure research needs are being met. And with that introduction, please allow me to pass the presentation over to Dr. Taylor.
Oh, well, thank you for that introduction, Michael, and thank you all for joining us here today. I'd like to start out by just giving a brief overview of what I'm going to cover with regards to in vitro MetID. I'd like to first talk about the role of in vitro MetID in drug development. Then we'll briefly talk about some definitions that are used when related to MetID studies, and talk about what the regulatory standards are for in vitro MetID. We'll then go into the timing of in vitro MetID studies. We'll talk about the various in vitro MetID test systems that are available, briefly touch on in vivo studies, and then talk about the quantitative information that can be attained from in vitro MetID and the in vitro MetID endpoints. So in general, when it comes to drug discovery and in vitro MetID studies, the guidance for industry and the safety testing of drug metabolites, or MIST guidance, encourages the identification of any differences in drug metabolism between animals used in non-clinical safety assessments and humans. And the overall message from the regulatory agencies is that this work should be conducted as early as possible in the drug development process. Some of the things that the regulatory agencies are looking for are how many metabolites are formed and what are these metabolites. Also, they want to know, are there any human specific or any human disproportional metabolites compared to any of the tox species that are being evaluated. Now, the reason that the regulatory agencies want this information is because metabolite profiles can vary across species, both qualitatively and quantitatively. Additionally, many of these metabolites may be potentially pharmacologically active and or chemically reactive. Now, so not only is this a box-checking exercise for the drug developer, but getting this information as early as possible can also provide some real benefit and value. This can inform some no-go or go decisions and also help give some information on to how to design future in vitro ADME DDI studies. Now, moving on to some definitions. Now, MetID is a very all-encompassing term that we use in the industry. However, there are more specific terms that should be used that have more specific meaning. So what do we call this? Is it metabolite profiling, metabolite characterization, or metabolite identification? Each of these terms has some specific definitions about what's involved. So the first would be metabolite profiling. And metabolite profiling is kind of the simplest of the MetID steps. And metabolite profiling just tells you how many metabolites are formed in each species or test system. So for example, here in these chromatograms, we see that it's done with monkey hepatocytes and human hepatocytes. We see that there's a parent and that 2 metabolites are formed. Now, taking this to the next level would be metabolite characterization. Now, metabolite characterization allows for the determination of the molecular weight and elemental composition of a given metabolite. So, for example, we have a parent, and in this case, based on the change in its mass, we know that an oxygen has been added. So this metabolite's been oxygenated, but we don't specifically know where on the molecule this transition took place. So now in this same example with metabolite characterization, again, we have our monkey and human hepatocytes, we have the parent, but now instead of knowing, we know more that it's not just metabolite 1 and metabolite 2. In this case, we know that we have a glucuronide metabolite along with a sulfate metabolite. So we're getting a little bit more information about what these metabolites are and the biotransformations that have occurred.
Andrew,
yeah.
Sorry, just to quickly interrupt you. We did have some questions come through from our attendees prior to the webinar, and one of them was just about the software that you use for this structural determination. Is there any specific software that you do use?
Yeah, no, that's a great question. We do have specific software that we use when we conduct our metabolite characterization studies. We currently use the Waters Unify software for MetID and data processing. Now, this has the capability of biotransformation localization. It's based on the elevated energy, high resolution full mass scans. However, for our definitive studies, we do manual interpretation of neutral losses, and fragment ions in the MS spectra for the best structural elucidation of the parent and the given metabolites. So it's kind of a mix between using the software and then a manual overview and assignment of the metabolites. So great question. Okay, so moving on to the last one, metabolite identification. Now, metabolite identification is giving a definitive structural assignment to the transformations that have occurred to the parent. And this is typically accomplished by an exact match when comparing the retention time and the MS fragmentation with an authentic reference standard. Additionally, this can be done by NMR analysis using the definitive chemical structure. So for the most part, though, when it comes to in vitro MetID studies, they're mostly performed as metabolite characterization studies. However, again, if metabolite reference standards are provided, those can be used to confirm the identity as applicable. And as I'll get to later, if reference standards are available, they can also be used to quantify the amount of metabolites that are formed. So when it comes to the regulatory agencies, metabolite characterization studies, or in vitro, I should say, metabolite characterization studies, do not need to be conducted under GLP conditions. However, here at Sekisui XenoTech, metabolite characterization studies are performed as non-regulated studies, but all of our studies are conducted in accordance with the applicable standard operating procedures, or the SOPs of our facility. And all of these SOPs were developed based on the high standards of record keeping that is outlined in the FDA regulations, CFR 21, Part 58. So the timing of in vitro MetID studies. This is a question that we get asked often. When should I conduct these studies, and what version of an in vitro MetID study should I conduct? Now, as I mentioned earlier in this presentation, these studies should be conducted as early as possible in the drug development process. For example, cross-species in vitro MetID studies are conducted early generally to assist with tox species selection. Also, in vitro MetID should be conducted early in order to determine if there's any potentially toxic or reactive metabolites that are formed. In addition, conducting these studies early, say prior to reaction phenotyping studies, can give some guidance on how to design those other studies later on. So for example, if we see that a lot of our metabolites are glucuronide metabolites, that's going to give us a good indication that we're probably going to have to conduct a UGT reaction phenotyping study in addition to the CYP reaction phenotyping studies. So test systems that are used in vitro MetID studies. Selecting the appropriate test system to use in your in vitro MetID study is a very important first step. So cryopreserved hepatocytes are the most commonly used test system for in vitro MetID studies, and they're kind of considered the gold standard to use. And that's because intact hepatocytes contain all of the major hepatic drug metabolizing enzymes, including the enzymes for hydrolysis, reduction, oxidation, and conjugation. Additionally, hepatocytes, since they're intact living cells, already contain all of the necessary cofactors that are required for many of these drug metabolizing enzymes to be activated. However, there are other options that can be considered. For example, subcellular fractions such as microsomes or S9 might be chosen. One reason to choose a subcellular fraction is if maybe there's a concern that the compound of interest might not be able to enter into the cells. So in that case, a subcellular fraction might be a good choice. However, when using subcellular fractions, since they're not whole cells, these need to have the appropriate cofactors added to them. For example, NADPH for CYP enzymes or UDP-GA for UGT enzymes. But the nice thing is that these can be added or withheld as needed, depending on the questions that are being asked. Additionally, attachable or plated cryopreserved hepatocytes could be a good choice if the compound happens to have low turnover, as attachable and plated hepatocytes can be incubated for a much longer period of time. So choosing the appropriate test system is something that needs to be taken into consideration. So metabolite characterization of in vivo samples can also be conducted, but I'm just not going to go into too much detail on this, as Heather is going to talk about this later on. But in vivo samples from preclinical and clinical studies can also be used from various tissues. So quantitative metabolite characterization studies. Generally, MetID studies are not quantitative. They're conducted more as a qualitative assessment. Now there are some exceptions to this. So unless it's a radioactive, radio-labeled compound, if it is, then we can conduct quantitative studies. And radio-labeled studies will provide quantitative information for each metabolite that's formed. Now the caveat to this is that the label needs to be retained on the metabolite. So when choosing where to place the label, some thought needs to be put into that ahead of time. So why are metabolite characterization studies not quantitative? Now, the reason for this is that mass spectral data are not reliably quantitative due to the inherent differences in ionization efficiency between the test article and its metabolites. So there's an example shown here. So if we have three metabolites that have the same biotransformation, this hydroxylation, however, we can see that it's put into different positions on the ring. So either in the ortho, the meta, or the para position. Now, if we look at just the peak height or, of these metabolites, and if they're put in equal molar concentrations, we can see that the ortho and para isomers have very similar peak heights. However, the meta isomer is about double those of the other two. And this is, again, just simply due to differences in the ionization efficiencies of these isomers. Now, there is a kind of a middle ground. If we look at the peak height of the UV signal, it's a lot more similar. So as we can see, we have 1.1 times 10 to the negative 2, 1 times 10 to the negative 2, and 9.5 times 10 to the negative 3, which is essentially 1 to the negative 2. So if we're lucky enough to be able to get a UV signal from the analysis, that can be used as kind of a middle ground or a semi-quantitative way to look at the metabolites. And again, the other way to quantify them would be to use a radio-labeled compound, and or if reference standards are available for the metabolites, then of course we can quantify them. Now, what are the endpoints that come out of an in vitro MetID study? The endpoints include how many metabolites were detected in each species, if it's a cross-species MetID study. We can give proposed biotransformations that led to their formations, and also any putative structures of the metabolites based on these biotransformations. We can determine if there are any human-specific metabolites, or if any of the metabolites were detected disproportionately in humans than other species. Additionally, the information provided will include the UV peak areas for the parent and its metabolites. And lastly, and again, the proposed metabolite structures and fragmentational assignments can be proposed. So here's just an example too of why a cross-species metabolite, in vitro metabolite identification study is important. If we look at the compound coumarin, in humans, it is metabolized into 7-hydroxycoumarin, which has no toxicity to it. However, in rats, coumarin is metabolized into this epoxide form, which is highly hepatotoxic. And this is just a table of an example of the data from a cross-species in vitro MetID study. We can see in the various rows, it lists the components or metabolites that are formed, along with their retention time, mass shift, and the proposed biotransformations that occurred. In this example, this compound was tested in mouse, rat, dog, pig, and human, and we can see that metabolites 1 through 5 were formed in all of these species. However, metabolite 6 was only formed in pig and human. So in this example, when selecting a non-clinical tox species, pig would be the better choice as opposed to mouse, rat, or dog, just because it has the most similar metabolic profile to human. Now that's all I've got here for in vitro metabolite identification, and I'd now like to pass it off to Heather.
Thanks, Andrew. Just before we move on to the bioanalytical portion, we did have another couple of questions that I think fit into what you've just gone through. So one of our attendees talked about what happens if a unique metabolite is observed in clinical studies and not in the non-clinical studies, and what can people do to address this? And also proactively prevent it from happening.
Yeah, that's a great question. I'm going to answer the second part first, and I don't think that there is any way that to prevent that from happening. I mean, this is in vitro MetID. These are in vitro studies. Therefore, it's not always going to be completely representative of what happens in vivo. That being said, I think there are a few things that you can do to maybe help mitigate that, and that would be the selection of the appropriate test systems. So, for example, selecting hepatocytes as the test system, something where all the metabolic pathways are present, and active is probably going to give you a lot more similar results to what you're going to see in the clinic. But again, it's impossible to always get the same results in vitro that you're going to see in vivo or in the clinic.
Absolutely, thank you. Another question that I have, not from the attendees, but you talked a lot about the different stages. Is there any kind of trend on where you see clients coming to you at what stage during their program they're getting this work done?
Yeah, again, great question. So we see and we get requests for in vitro MetID studies kind of across the board. We generally do get them a lot in the early stage, and those would consist of the cross-species types of studies when they're looking for the appropriate test system. However, sometimes we do get requests in later stages. Maybe something was seen in the clinic that wasn't expected, and they might come back and repeat a MetID study in vitro. And then, of course, in the middle, because the guidance doesn't exactly specify when this needs to be done. So sometimes it's just done in the middle. It all comes down to where the sponsor is in their drug development process, how much prior information they already have, and what de-risking steps that they want to take.
Great, thank you very much.
You're welcome.
So I will jump into the bioanalytical portion of the metabolites work and talk on some key topics and talk through some questions that we're commonly asked in the bioanalytical space. So I'll speak through the points here. So looking through metabolite analysis and bioanalytical, some different options that we routinely see for that analysis. And if you're in a situation where you're not doing metabolite analysis, just a little bit of information on what is most helpful for the bioanalytical laboratory to know. And then I'll talk through just some examples and case studies of where we've seen metabolites and analytes throughout various stages of a clinical and also preclinical programs. So linking to what Andrew has talked about, it's quite similar. We see varying amounts of metabolite analysis throughout the bioanalytical phases. So the extent of this assessment can be dependent on a variety of factors. I've outlined some of them here, but it comes down to predominantly how much information is known about the analyte and also the metabolites, whether those metabolites are active and therefore need to be quantified in the clinical and preclinical space. Can also depend on the type of drug that it is. If the parent analyte is not actually the active one, again, it requires that that metabolite would need to be analysed. It can also depend on the current phase of the program. If you're looking later phase, you're likely to have more information about your metabolites and therefore be looking to quantify them. Something we do often see is that the metabolic pathway can actually be different from a genomic point of view, such that we need to be looking at fast metabolizers and slow metabolizers to better understand that pathway. So if the preclinical or in your MetID testing shows that you do need to be monitoring your metabolite as you progress through your drug program, we've seen that done in various ways, and it can largely depend on the requirement of that data, such that is it going to be used for safety testing? Is it going to be used as exploratory just to kind of look what's there? Or is it going to be used for the fully regulatory package? So I'll talk through these 3 points in a little bit more detail, but the analysis of metabolites can range from monitoring of the metabolite, measuring of the metabolite using a fit-for-purpose assay, and measuring of the metabolite using a fully validated assay. So it is worth noting at this point that each metabolite, as per the regulations, does have to be treated as a unique analyte. And while some analytical methods may be combined to contain the parent analyte as well as multiple metabolites, each of those analytes does need to be treated individually against the required acceptance criteria.
Heather, can I interrupt for a second? I have a question.
Yeah, absolutely.
On the last slide.
Mm-hmm.
So at the end, you say that the regulatory guidance requires that each metabolite is treated as a unique analyte, but yet analytical methods may be combined. Is there any sort of limit on how many of those methods can be combined into one, or what plays into that?
Yeah, it's not a nice answer, unfortunately, but the answer is it depends. So there's not necessarily a limit to the amount of analytes that can be included in an assay. But as more analytes are included, it becomes more complex from an extraction point of view and also a data interpretation point of view. It also does rely on the analytes being chemically similar enough that they can be included into the same assay, such that we can produce that really robust assay for all analytes equally. So sometimes it's actually not possible to include them all together, and it provides the best data to have separate assays.
Gotcha. No, that makes a lot of sense. Thank you.
No worries. So moving on to discuss in a little bit more detail those 3 options that I mentioned earlier. So probably the most non-regulatory and most exploratory way to look at your analytes would be purely monitoring of the metabolite. So this would be very exploratory in nature, and you'd expect there'd be no clinical endpoints, regulatory or safety decisions made from this data. There'd be very limited scope and robustness to this, and it involves just monitoring of the MRM transition, and you may not have any reference material available for this stage. So while this is possible, this is probably more in the MetID space, and what we more routinely see is the next option, which is measurement of metabolites using a fit-for-purpose assay. So the FDA guidance includes the concept of a fit-for-purpose assay. This allows for the level of validation to be appropriate for the intended purpose of that assay. and of what you're looking to do with that analyte data. So for this in order for this option, we would need a reference material to be available, and for full characterization of the metabolite to have occurred. So as Andrew spoke about earlier, we would need to know that full profile of that metabolite. But the difference here is that limited validation is performed in a fit-for-purpose manner, which may not include all of the testing, but gives that indication to the client as to how the metabolite is behaving without necessarily going to that fully validated space. So this again would be for exploratory clinical endpoints, and we wouldn't expect any regulatory or safety decisions to be made from this data. It would just be to have a look in a little bit more detail how the metabolite is performing in either the preclinical or clinical studies. So moving on to the next option, being measurement of the metabolite using a fully validated assay. So this would be fully validated to the standards of the FDA and EMA guidances. Again, it would have required that full characterization of the metabolite has occurred and that reference material is available. And this could be used for clinical endpoints and safety decisions. So this would be used when your MetID or preclinical data has indicated that the metabolite needs to be assessed for safety reasons. So there's some common examples of the different ways that metabolites can be analysed in the bioanalytical space, but there is also the occasion that you don't need to monitor your metabolites as indicated through your MetID testing. So if this is the occasion, we often get asked what info do you need about metabolites to do what you need to do for the parent? And the answer is, put plainly, as much as possible. So as much information as you've got available is helpful in developing the assay for your primary analyte. And the reason for this is that it can help to identify potential analytical concerns early on in the method development, such that we don't have to wait to get subject samples potentially in order to see these concerns to be able to fix them. Two of those which come up in this space is about stability. So some metabolites can often be quite unstable in that they break down and reform the parent analyte, or they may themselves break down to form another entity. This can result in a analytical result for the analyte, the parent analyte, which is not reproducible because that metabolite is breaking down and impacting your analyte quantitation. This can often manifest itself by failed ISR data, so incurred sample reanalysis. And by that point, you're quite a way through your human program, and that would require the assay to then be redeveloped, looking at what has caused that instability. Similarly, from a selectivity perspective, moving through into the subject samples where those metabolites actually are is the first chance that we'll be able to see if they do actually interfere. So if we have the data available to us, then we're able to have a look at it and see if there's any potential in that. So both of these issues, if they do come about in the human analysis, they would require the potential repeat of the development and validation, resulting in a loss of time and money for the client. So a lot of these issues really come down to a risk assessment, in that if we go ahead at this stage versus delaying it, there could be a potential need to repeat some work. But again, with any risk, that may not eventuate. So similar to what Andrew went through, if I don't need to monitor my metabolite, when do I need to? And the FDA does say as early as possible in the drug development process. And again, from a risk perspective, that is a better option because you're likely to be able to cover off any concerns early on in your program. Notably, it is quite frequent that the metabolic pathway can differ between non-clinical species and with humans. And as per the FDA safety testing of drug metabolite, if a human metabolite is present at greater than 10% of your total drug-related exposure, this is required to be assessed for safety. So that's just a little bit of information which really kind of links into what Andrew's been talking about as also. So if it came about that you had started your human trials and then needed to include a metabolite after you had started, as your information available and you do more testing as you progress through your phases of your program, what options are available to you in that space? So it broadly falls into two categories. So you've already got collected clinical samples. So those clinical samples could be reassayed for this metabolite. That would depend on the collection conditions being comparable. So as I mentioned before, sometimes the metabolites can be unstable. If the assay hasn't been developed and the clinical collection conditions haven't been put in place specifically for that instability, you could find that you have to recollect the samples with specific stabilizers included. In that situation where you may not be able to use the samples already available to you, it may require the studies to be repeated to collect that data. So I'm just going to talk through some case studies, some examples of varying, where we have seen varying degrees of metabolite analysis throughout preclinical and clinical programs. So one example I've got here is where a metabolite was identified prior to the commencement of the preclinical studies by the client in the work that they had done in that MetID space. Due to that, we were able to develop an assay in rat and dog plasma, and it was observed that the metabolite was very unstable in dog blood. So we had to use additives to stabilize that metabolite and put in place some fast processing of that blood to make sure that that metabolite, when it was analyzed, was representative of when the sample was collected. If we didn't have that data available to us, and if we weren't looking for that metabolite, then it's potential that we would have collected those samples in a very different way, and then not have been able to use those samples to measure that metabolite. As the client progressed through this program, they performed additional MetID on the dog samples and identified another metabolite which was required to be incorporated into their human assay. That actually required a custom blood collection tube due to the instability of all the metabolites to make sure that the most robust data was produced for those human studies. So a different example was where we had performed phase 1 studies for a drug program. And in the late stages of their phase 2 and phase 3 trials, it was determined that a metabolite was required to be analysed. We developed an assay for those metabolites. Luckily, the analytes were stable in the way that the samples had already been collected. So we reassayed these samples from the phase 1 trial for that metabolite. Due to the time difference between when those samples were collected and when they were analysed, was required to prove retrospective stability for those metabolites for up to 3 years. Another example is where we've seen it's been identified that a metabolite has been required to be analysed in the early stages of the human first in human trials. In that case, the assay was updated early in that trial to include the metabolite into the existing assay. So that's an example where it was able to be one assay. We validated for that assay, and then analysis continued with the new updated assay throughout the trial. So that concludes the presentation portion. Thank you everybody for listening. I have another couple of questions for you, Andrew, if that's okay.
Oh yeah, no, and I for you as well. So
you go first. I've asked a lot.
Sure, of course. Well, not necessarily a question, kind of more of a statement, but one thing that's well, first of all, coming from the in vitro side and learning about the clinical side, that was a great presentation. Thank you, by the way. I learned a lot. But seeing the similarities between the in vitro side and you from the clinical side on what's required. I didn't go into things that we generally ask for from the sponsors, but for in vitro as well. I saw, you brought up having as much information as possible from the sponsor and how important that is. That's always something to remember as a sponsor. Things like structure, stability, any sort of information, methods, those sorts of things.
Yeah.
Is there anything else you'd add to that from the clinical side, for the work you do that you like to have up front?
Yeah, a lot of moving into clinical, as much information as the preclinical testing that's been performed. Any opportunity that we can get to get ahead of potential concerns or potential problems is always better, because it always saves us and the client money and time. So any thing that was observed during the preclinical, as far as the bioanalytical portion, any instability, really important to try and make sure that we're developing the best assay for that human study. So from a bioanalytical perspective, it really is, as much as there is available, is good to receive.
Right. Makes sense. That's great.
I obviously have the bioanalytical background, so MetID is quite new to me. Do you find that a lot with your clients as well, that there's a bit of a back and forth in the understanding of what's required?
Yeah, absolutely. And generally, if someone's coming to us with, say, a toxicology background, I kind of feel like, at least when it comes to the cross-species work, that they don't have as many questions. But others, yeah, we generally do get a lot of questions. Some of the most common questions are, can this be quantitative? Can I make comparisons of the metabolites that I see to one another, just, and compare it to the parent? And again, the answer to that is no, unless reference standards are employed or it's a radio-labeled compound. That being said, however, you can make some comparisons of a given metabolite. Oh, and there's my kitty. A given metabolite. She likes to join webinars. But you can make comparisons of like a metabolite across species. So if we just look at one metabolite, metabolite number 2, we can use the peak height to make comparisons then across the species for that one metabolite, just under the assumption that the ionization efficiency of that given metabolite is going to be the same regardless of species. So that's probably the most common question, is about the quantification.
Okay. Great, thank you.
Yeah, I also I have one more that came through earlier, and the question is, can in vitro MetID be used to reduce in vivo tox testing? And that's a great question. And I'm not from the tox side, but my guess would be no, that there's really nothing that can be done. I mean, you're still going to have to do what's required by the regulatory agency when it comes to toxicology testing. However, what it can be used for, and like I said, is to assist in selection of the appropriate tox species, and also just give you some information up front that's going to help design future experiments and those sorts of things. So great question. And I think that's all I have on my end.
Yeah, that's all I have on my end. I think that's probably wrap up.
That was great. No, I really enjoyed working with you on this and learning about the other side of the drug development process.
Yeah, no, same. It was great to learn more about MetID, because it's not, it's fantastic.
Yeah, well, I guess thanks everyone for joining, and contact either one of us if you have any questions.
Thank you, Heather and Andrew. If there are any further questions, please feel free to submit them in the post-webinar survey or contact us through our websites, where you can also find a variety of information related to our services. Agilex Biolabs, the Australian specialist bioanalytical and toxicology laboratory facilities for clinical trials, has more than 25 years of experience in performing regulated bioanalysis, including quality method development, method validation, and sample analysis services globally. XenoTech specializes in ADME-PK and drug-drug interactions, and likewise has more than 25 years of experience in providing in vitro and in vivo research services, as well as cell and tissue-based products globally. Further resources on ADME and DDI, including papers, posters, other webinars, and more, is available at XenoTech.com. Have a wonderful rest of your day.